Okay, so now we're in the home stretch. We have 3 more talks, and we're really excited to have Dr. Yi Ming, who will be speaking to us today. Dr. Chen is a rheumatologist and also the director of many things at the Taichung Veterans General Hospital. And among the things that he's leading is the precision medicine effort there. And Dr. Chen was trained at National Yangming University, and then he did a lot of stuff, and then he spent some time at the NIH, according to his CV. So we're very glad that he's going to tell us about real-world journey of precision medicine in the hospital. Welcome, Dr. Chen.
Thank you, Puyi. Good afternoon, everyone. Today is my great honor to be here on this podium, standing with several outstanding speakers from abroad. And today I'm going to share with you our hospital experience using the TPMI data. As you may know that the Taichung Veteran General Hospital, we have enrolled more than 100,000 participants, and we genotype more than 60,000 participants. So we linked the genotype data to the EHR record. So the first one I'd like to share with you actually is called the alcohol flushing syndrome. You might have the chance to see some of the red faces tonight during the dinner. And actually the alcohol flushing syndrome is quite common in the Asian country. It is related to 2 genes. One is ADH1B and the other one is the ALDH2. Actually in Taiwan, we have a very high prevalence of ALDH2. The prevalence rate would be around 45%. So 1 in 2 people drinking in Taiwan may appear alcohol flushing, including me. And we have the privilege to link the 2 genotype data to our electronic health record. And we find out that with both these 2 genes, genetic variations, the patient may develop a higher risk of cancer, especially the esophageal cancer and the hypopharyngeal cancer. Why is that? Because the patient with these 2 type of genes variations, actually the alcohol cannot be metabolized well. It will lead to the accumulation of formaldehyde in the blood. And then it could lead to alcohol flushing and also carcinogenesis. So next time when you see a patient with alcohol flushing, maybe in addition to do a genetic test, maybe told him to avoid drinking more. And this paper is published 2 years ago by one of our ENT doctors. And the ENT doctors, he is quite confident with the TPMI data, but he says that can we do a lot of studies in the hereditary hearing loss? And then we look in the literature that the GJB2 actually is the leading gene of hearing loss in Taiwan. But in the previous literature, we don't have a large scale of publication from the Taiwan population. So we have the chance to look into the TPMI data. But very unfortunately, in the first version of the TPMI cheat, the GJB2 is not available. So we have to do PCR by our own. So we do a PCR for more than 6,000 men and 7,000 women. And we have the allele frequency from our hospital that the GJB2 V37I is around 0.92. And then the ENT doctor, they try to link the genotype data to audiograms in our hospital. So we can plot these 2 figures from the right ear and from the left ear. And on the x-axis is the frequency. And you can see the hollow box is the homozygous of GJB2. Actually, with the homozygous, you can see that at a high pitch, there will be a remarkable high pitch loss in those with homozygous, but not in the heterozygous, not in the wild type group. So we now know that GJB2 in Taiwanese is an autosomal recessive inherited pattern. And then we perform a phenotype-wise association study. Not surprisingly, we find that GJB2 is associated with hearing loss and also tinnitus, which is quite common in this type of patient. But to our surprise, we find out that fracture of humerus and different other skeletal sites pop out in the PHIOAS association study. Why is that? And then we look into the literature, we find out that the cranial nerve, the eighth cranial nerve, actually control hearing and also balance the vestibular function. So if next time you encounter a patient with hearing loss and maybe a family history of hearing loss, then you have to look into the gait disturbance. Maybe the patient may have a higher chance to develop fracture due to instability. So I think the genetic study from a hospital-based study may have clinical implications. And these two studies were from single SNP association studies. And in the morning, we hear from Eric that pharmacogenomics is quite important, but in the previous time, we just look in the literature. There's no publication from the Taiwanese population. So now we have a chance to look at the clopidogrel and CYP2C19 to see whether the pharmacogenomics has an impact on the outcome of a patient with coronary artery disease. So we work with our cardiologist, and we are able to link the genotype data to 1,000 patients who receive cardiac catheterization. And we have the number around 15% of Taiwanese that have poor metabolized for Plavix. So this number is quite large. So we can look into the database. We find out that in the 1,000 participants in the old data set, the pharmacogenomic data seems not to affect the outcome within 1 year after the cardiac catheterization. The outcome is restenosis and the cardiovascular death. But if we narrow down to those who receive cardiac catheterization due to acute myocardial infarction, then the PM phenotype will perform the worst compared with the normal metabolizer. So it appears that with our own data set, we know the frequency of PM and we know which population we should target. And the second study, we work with our pharmacist to look into the carotid catheterization because we think that maybe in the cardiovascular system and also the cerebral vascular system, the CYP2C19 may perform similarly. So we look into the number of carotid angiopathies. We find out that those with PM phenotype actually receive more times of carotid catheterization compared with those with normal metabolizer. So I think with the cardiovascular risk and the cerebral vascular risk, actually CYP2C19 is quite important and it affects around 1 in 6 Taiwanese in the population. The other one gene, pharmacogenomic gene, is TPMT. As a rheumatologist, I prescribe azathioprine at a daily basis for patients with lupus. And sometimes we will encounter patients who develop hepatitis in our clinical practice. And it is especially so true in Taiwan because we have a lot of patients who is hepatitis B carriers. And we put some effort for the differential diagnosis, what is the really cause of hepatitis. But in our hospital cohort, we identified that around 2.6% actually is a poor metabolizer of azathioprine. And we look into the longitudinal data, we find out that the accumulative event of hepatitis within 3 years actually is significantly higher in those with poor metabolizer for azathioprine compared with that with normal metabolizer. So I think this study is quite important even in the differential diagnosis for patients with hepatitis in Taiwan. And the hospital doctors, they say, not just do the study, what can you do for the patient? So we try to incorporate the pharmacogenomic information into our prescription system in our hospital. So we do a test run in the rheumatology division, and for it already for 3 or 4 months. But we ask the Academia Sinica, can we expand the whole the pharmacogenomic data to the whole hospital? And Academia Sinica says that we must go to the ethics committee first. So we applied for the ethics committee 2 months ago, and then we just get the approval last week. So we are going to enroll the whole pharmacogenomic information throughout our hospital. And next time, actually the genotype has been preemptively done. So next time when the patient who is in the TPMI cohort and the doctor prescribe a drug that is linked to CPIC guideline, that should be a warning. So our hospital will pop out a warning system in the prescription system. I think that can really help the patient and enhance the prescription safety. And the study I just reported is retrospective studies. Now I would like to introduce some of the prospective study. The first one is CADASIL, which is an autosomal dominant arteriopathy that can link to stroke. With the NOTCH3R544C mutation, the patient may develop premature stroke, and the risk is around 11 times. And the patient can accumulate hyperintensity on the T2 flare image on the brain MRI. In the pathology, there will be atherosclerosis and the stroke. But before the onset of stroke, patient may develop tinnitus, gait disturbances, and also memory loss. But we don't know when, at what age, would the carrier of NOTCH3 mutation may develop this kind of symptoms. In previous time, the neurologists, they did a lot of study. They compare a group of patients with stroke and the other group of patients without stroke. They find out that NOTCH3 is quite important. But now with the TPMI data, we have a chance to look at the carriers. We can see on their 20s, on their 30s, on their 40s, when would they develop hyperintensity in the brain, and how will this affect the symptoms and also the cognitive functions. So I would like to thank Puyi for his kind support, grant support for us to perform this prospective study. We know that in our hospital cohort, the NOTCH3 mutation MAF is around 0.96, around 1%. So we call back the patient with the carriers, and we have enrolled 150 patients with NOTCH3 carriers and another group of non-carriers. We compare them and we record the neuropsychiatric assessment. We also do a brain MRI. And then we are anticipating to follow up every 2 years for the clinical symptoms, for the neurologic assessment, and also for the brain MRI. And these 2 figures actually shows the correlation, the regression between age and the hyperintensity volume on the brain MRI. We use a quantitative method to calculate all the white spots on the brain MRI. And we plot it on the y-axis and also the hyperintensity lesion to whole brain volume as a ratio. And the green dot is a NOTCH3 carrier. The blue one is a healthy control. And you can see at the age of around 50, these 2 groups start to separate. So our study shows that maybe for those with NOTCH3 carriers at the age of 50, your brain start to accumulate hyperintensity lesion. And we are quite interested in, are there any factors that can also contribute to the hyperintensity lesion in the brain? The NOTCH3 variation is from the birth, at the day of the birth, but at the accumulate comorbidities. How would that contribute to the development of brain lesions? So we do a multivariable regression analysis. We find out that, of course, the age, with the advancing age, you accumulate hyperintensities. But also for the vascular risk factors index, hypertension, diabetes, hyperlipidemia, cardiovascular disease, and also smoking. All this can contribute to the white matter hyperintensity in our cohort. But just brain lesion, is that linked to the cognitive decline? So we do a a comprehensive cognitive assessment, it takes around 1 hour. And then we find out that with the increase of hyperintensity in the brain, that is shown in the y-axis, actually the general cognitive function in terms of MoCA, the memory function in terms of 12 item memory test, and executive function and processing speed in terms of digital span test actually decreased. The cognitive decline actually increased with the volume in the brain. So I think these brain regions actually means something. But as we discussed at lunchtime with Professor Zhong, what should we do? Is there any intervention for the carriers? Actually, we don't know the answer. But fortunately, our neurologist, Dr. Li Weizhu, he has the grant support already from the NSTC for 3 years. So we might have the chance to conduct a prospective genotype-based clinical studies to see whether the interventions, the antiplatelet drugs for the NASH3 carriers in CADASIL cases. As a rheumatologist, I take care of patients with systemic lupus. This is our team's previous work. We find out that in the fluffy babies, the Afghan hunt, he may develop skin lesions just like patient with lupus. It's just like discoid rashes. And we have a chance to look at the dogs raised by patient with lupus and then compare with dogs raised by patient without lupus. We try to look at the RNA positivity in these two types of dogs. And we find out that dogs raised by patient with lupus actually has a higher signal of autoimmunity. How is that? This is not a transmissible disease. The SOE is not a transmissible disease because of the environmental factor, we guess. And maybe due to the virus infection or the environmental exposure, UV light. And as you may know that patients with lupus, they typically have the rashes on their faces, and it is linked to sun exposure. So with the TPMI data, we can calculate the genetic background of the patient by polygenic risk scores. We use our cohort to establish a PRS for SOE, and the component gene is around 27 genes. And within the 27 genes, we find out that EST1, STAT4, TNF-AIP3, these 3 genes has been reported to be associated with photosensitivity in the previous publications. And then we have the PRS score for every patient with lupus. Then we have a chance to look at the development of SOE among our cohort, which is shown here in the red curve. On the x-axis is the SOE PRS percentiles. You can appreciate that with the increase in SOE PRS percentiles, the risk, actually the risk of SOE development is getting higher. But not everybody gets SOE, because in the highest SOE PRS percentile, around 10% of them develop SOE. But we have a chance to link the patient's home address to the Central Weather Bureau sun exposure database. So we know that the exactly index day of SOE onset, 1 month before that day, 1 year before that day, 2 years before, we know that the solar exposure data before the development of SOE. And we try to figure out what is the gene and the environmental interaction between SOE and the sun exposure. To our surprise, that we find out that with the increased SOE percentiles, PRS percentiles, actually the dose need to develop lower. So if you had high SOE PRS, then you are sensitive to UV exposures. And then we separate the patient's sun exposure to a low solar insolation and high solar insolation. If you are in the low solar insolation group, even you have very high SOE PRS, you will be fine. You will be okay. But if you are in the high sun exposure group and you have high SOE PRS, then you will be in trouble. But I realized that the error bar is quite large, which means we have to accumulate more data. And this manuscript actually has been rejected this morning. So we have to find out somewhere to get it published or maybe accumulate more data. And I think this data may have a clinical application for patient who lives in Taiwan, because actually we are a tropical country. The sun exposure is so remarkable. And I am frequently asked by patients with SOE, how can I do to prevent SOE from happening in my daughter? Now with this data, I may have more evidence to talk to the patient. You should do the PRS first. If your PRS is quite high, maybe you should not go to the beach. You should not go to the sunbathing. I think that's the clinical application. And we also have a chance to use machine learning in our our hospital database. When you get the SOE, the rheumatologist is interested in when you will have lupus nephritis. And then we use our hospital database around 1,500 SOE patients. And we use SNPs, the PRS, and the electronic health records, and use machine learning model, try to build up the prediction model. And we find out that with both the integration of PRS and the electronic health record actually predicts the best the development of lupus nephritis. And the AUC is around 0.89. And the last part of my talk would be what happened to the immune system before the onset of SOE. Now we know the SOE PRS in each participant of the TMMI. And we have a chance to call them back and to do a blood test. But we decide to call back female participant aged 20 to 50 because at this age and this sex is a high risk for SOE. So we call back top 5% of participants with SOE PRS, and then we draw a bunch of blood from them. And we compare them to another group who has low SOE PRS. And we find out that the nucleus antibody, which is a screening test for autoimmunity, is quite high. In 1/3 of the high-risk patient, they have positive ANA higher than 1 to 160. But in the normal cohort in this age group and female, it is around 5 or 6%. So with the SOE PRS, before the disease onset, you might have autoimmunity going on in your body. And we have the chance to see other immunologic criteria, for example, low complement, anti-double-strand DNA antibodies, or anti-phospholipid antibodies. A substantial number of them already have the autoimmunities. And we try to find out what is the pathway, the differentially expressed genes in those with pre-SOE. So we do a single-cell RNA sequencing. Actually, we don't know how to do a proper analysis, so we just at the infancy of the data analysis. And we think that in patients with SOE, they produce a lot of autoantibodies. So the B cells must be very important. So we just look at the B cell in the single cell data set, which is shown in Tiffany blue. And you can see the B cell numbers and the monocyte increased in the pre-SOE group, which is the high PRS group, and also the SOE groups. So we look at the B cells. But you can also observe that in monocytes, the number in pre-SOE and SOE also increased. But we don't have the time to do the analysis yet. So we just look at the B cell to see whether there are any differentially expressed genes. And we find out that PS26, which is a ribosomal protein S26, is increased in B cells in the pre-SOE group and also the SOE groups. And RPS23 has been reported in type 1 diabetes, IgA nephropathy, and a bunch of autoimmune disease. And then we have the chance to separate the patient into pre-SOE, only one criteria for SOE, and higher than one criteria, with ANA positive and negative, with low complement C3, C4 or normal complement. And you can see there's a dose-dependent relationship between PS26 pre-SOE and SOE compared with patient with healthy control. So I think with this approach, with the PRS and with the callback mechanism, we have the chance to look what's going on in the immune system before the disease onset. Actually, in rheumatoid arthritis, there has been publications showing that targeting B cells can delay the onset of rheumatoid arthritis. I'm also wondering, can we do that in the patient with pre-SOE? I'm not sure. We need to find out. And this is the last slide of my talk. I would like to thank my whole team of precision medicine in our hospital, the support of our vice superintendent, and I would like to thank Professor Xu Huiren for his support in the past and in the future. And we have the bioinformatics support. Dr. Xiao, he is a key leader of our team, and he's in the floor. And this is the bioinfo team. We have Dr. Zheng, who is specialized in cancer genomics. We have an epidemiologist. We have 2 talented, outstanding pharmacists. We have clinical laboratory technicians. We have the clinical informatics and the doctor who are taking care of animal models, and the younger generations doctors, because I'm going to retire very soon. So we have to cultivate our next generations. With that, I would like to thank you for your attention.
I have a question from the very beginning of your talk and a question from the very end of your talk. The first question I had was, in the case of the flushing that you described with the risk for esophageal cancer, if those individuals do not drink at all, does the risk for cancer go to 0?
We don't have the chance to look at data because in the hospital, the drinking, social habit, drinking, smoking, actually is not that reliable. So if we want to look at this signal, we have to do a prospective study.
Okay. I mean, although that would be very interesting to know, obviously. My last question, I was going to ask Pui at some point, I'll just ask now, is genetic counselors, is that a profession in Taiwan? And if not, has there been discussion about creating that as a career track?
I believe that is very, very important.
No.
Doesn't exist here.
Well, but we are going to get it started. And I have a question about the very beginning also. It's about the pharmacogenetics thing. You put the data into the medical system. You said that you just got IRB approval.
Yes,
but
last week.
This is not a clinical test. Did you get FDA approval?
No, this is not a clinical test. This is just a provider of genetic information, the study result. So we claim that this is not a genetic test. So we don't need to get through the whole approval process.
Okay, so I think that's going to be important sometime.
Building on that question, just it's a lovely, elegant way of getting the data back to the participants. Wonderful. But are you not worried about sample mix-up or admixture that might lead to the incorrect result appearing in the relevant medical record? What's your, say, sex swap rate in Taiwan Biobank when you come to do the genotyping?
Dr. Lee, what is our swap rate? A good guess. Okay, we'll talk about it at dinner.
Thanks for the wonderful talk. And actually, I have 2 questions would like to ask you. So the first question is about your finding, identify a risk factor about the sun exposure for increased risk of developed SLE. I just wonder, is the risk factor has been reported in other ethnicity population?
Let me answer your first question first. It has been a well-known risk factor for patients who develop SLE, then get worse with the disease flare. After the disease onset, UV light exposure has been a well-known risk factor, but not before the disease onset. So now we have the chance to look at before the disease onset.
And then, so it's just my own opinions, because I think for the ongoing manuscript you just mentioned, it just be rejected this
Right.
morning, and because probably limited sample size, would it be possible can establish a cooperation with Dr. Chamber and use the Singapore precision process project to kind of increase your sample size? That's just my own personal opinions. And my second question is that for you to share with a very wonderful pharmacogenomics result and then implement into your clinical practice. Would it be possible? Because I'm not familiar with our governmental department or section. I just wonder, would it be possible we can initiate a genetic screening? When is the mature timing for us to implement our pharmacogenetics finding to the genetic screening from a public perspective? Thank you.
I'm not, although I'm working in the public hospital, I cannot stand for the government, but there are a lot of private sectors. They are already doing the pharmacogenetic testings. A lot of them already came to me and they would like to have their product in our hospital list. And then I talked to the physicians, I talked to cardiologists. They say that we don't need the pharmacogenomics data. For the statin myopathy, I just prescribe the statin and the patient came back. If everything is okay, then we don't need the test. So I think maybe we need to education, more pharmacogenomics literacy for our medical staffs before application of the pharmacogenomic test.
Great. Well, thank you so much, Dr. Chen.